everyone, my name is Katalin Doneanu and I'm a principal scientist in the Biologics Biopharma organization at Waters. Today I will talk about some of the recent new informatic products developed at Waters for RNA sequence confirmation using the bottom-up enzymatic digestion approach. RNA mapping involves the enzymatic treatment of mRNA molecules to produce digestion products. One of the challenges that our customers are facing today is to generate unique, unambiguous digestion products from their mRNA molecules. In response to this challenge, Waters has recently launched two new digestion enzymes capable to generate unique digestion products through a combination of novel cleavage sites and miscleavages. Another customer challenge is to rapidly identify the digestion products in order to map the full RNA sequence. The new workflow introduced here enables better RNA sequence confirmation in a quick and streamlined fashion, therefore increasing the efficacy of drug development and the release of RNA-based products to the clinic or to customers. While oligomapping is predominantly used in research and development, our integrated workflow enables the potential use of this workflow in manufacturing and QC laboratories. Here is the end-to-end workflow for RNA mapping. It starts with sample preparation using the new Waters Rapizyme RNases and is followed by data collection using UPLC-TOF or QTOF system, BioAccord or Xevo G3. A newly launched software, MapSequence Waters Connect app, with two updated micro apps will be the focus of today's talk. mRNA Cleaver is a micro app used for in silico generation of RNA digestion products produced by a variety of ribonucleases. MapSequence is used for matching the predicted digestion product against the experimental data. The other micro app, called Coverage Viewer, is used for displaying the RNA sequence coverage based on the MapSequence results. Currently, the MapSequence app is using only the MS1 data channel with accurate mass analysis. I'm highlighting here the new sample preparation tools recently introduced by Waters. We launched two new enzymes that have unique cleavage sites, dinucleotide motifs, a very different cleavage pattern from the G-based specificity of RNase T1, which is one of the enzymes frequently used today. These two enzymes are Rapizyme MC1 and Rapizyme Cusatibin. MC1 cleaves at the 5' side of uridine residues, with three primary cleavage sites at AU, UU, CU, and secondary cleavages at CA and CG motifs. Cusatibin has 3' cytidine specificity, cleaving primarily at CA, CU, CG, and UA motifs, with secondary cleavages at AU, GU, and UU motifs. The unique specificity combined with miscleavage properties makes these two enzymes generate digestion products with longer sequences. These two enzymes reduce the number of short, unwanted digestion products like single, di, or trinucleotides and increase the number of longer digestion products, which have a higher probability of having a unique mass. I would like to provide a brief overview of the functionality within the mRNA Cleaver micro app. First, we paste the RNA sequence into the sequence box. In this example, we have an 100-mer single guide RNA containing 3 modified residues at the 5' terminus and another 3 residues at the 3' end. The 3 X residues present at the 5' terminus represent 2' O-methyl phosphorothioated adenosines, while the 3 Y residues at the other end of the molecules are 2' O-methyl phosphorothioated uridine. There is an expanded range of digestion enzymes available, including a list of 8 enzymes. In this example, we'll choose Rapizyme MC1. There are 4 possible options available for defining the 3' terminus of the digestion products, including either linear or cyclic phosphates, a combination of both, or a dephosphorylated terminus. In this example, we'll go with the enzyme default terminus, which for MC1 is cyclic phosphate. Furthermore, digestion products with miscleavages or containing predefined mRNA modifications, for example cap structures, can be included as well. In this example, we'll go with 4 miscleavages. One of the additional settings provided includes the minimum and maximum length of the digestion products. We recommend to leave the filter duplicate sequences box unchecked. In addition, a wide range of charge states is typically selected, in this case 1 to 10 charge states, negatively charged precursors, along with the M/Z range, which usually corresponds to the acquisition mass range. After selecting the processing parameters, we click on Calculate to produce a CSV file containing the predicted digestion products. In the mRNA cleaver, it's easy to add modified nucleotides with new structures, as well as to add new digestion enzymes, including enzymes with multiple cleavage motifs. These capabilities illustrate the flexibility of this micro app. After we created the predicted digestion products, I will show you how to create a processing method in the Map Sequence Waters Connect app. First, we enter the name of the processing method, in this case MC1 digest. There are only a few processing parameters required: the mass error, 15 ppm in this example, the minimum isotopic response, 1000 counts, and the peak detection window from our chromatographic runs from 0 to 35 minutes. The predicted digestion products from mRNA cleaver are then uploaded. In the injection list settings section, we select one or more individual runs that were previously acquired. In this case, we have only one injection. This is all that's required before we start to process the data. Data processing typically takes 1 or 2 minutes for single guide RNA digests and 5 to 6 minutes for mRNA digests. You can see the results table here displaying digestion products that have been found, highlighted with green circles. Those who have not been found, showed next to red squares, and those which are ambiguous, denoted by amber triangles. The table contains a lot of details available to review: the position and the sequence of the digestion product, is expected and observed neutral monoisotopic mass, along with the mass accuracy associated with this measurement, the MS signal response, and the retention time of the digestion product. There are 3 categories of ambiguous products, each highlighted by a different type of amber-colored triangle. They are ambiguous products produced by multiple chromatographic peaks matching the same target mass. These are called alternative peaks. There are also multiple isomeric sequences that could be assigned to the same mass at the same retention time, called alternative product assignments. In addition, it is possible to have both alternative peaks and alternative product assignments. The digestion products can be grouped by type, which sorts out the unique sequences from structure isomers. So here are examples of unique sequences, and down at the bottom of the table we get several examples of structural isomers. In the later case, there is enough information provided in the details section that allows us to investigate beyond the MS1 data analysis provided by Map Sequence. Using the Confirm Sequence Waters Connect app, it is possible to confirm the presence or absence of structural isomers found by Map Sequence using the fragmentation data available in the MS2 data channel. Finally, we can export the results table as a CSV file using the Export All injection button. The last step of the oligo mapping workflow is to use the Coverage Viewer micro app to visualize the sequence coverage. Here, we are entering the single guide RNA sequence in the sequence box, and then we select the mRNA cleaver CSV file containing the predicted digestion products. At this point, we can see that the predicted theoretical coverage for Rapizyme MC1 enzyme, which is shown in blue, is indicating that full sequence coverage is possible. Then we import the map sequence results, the CSV file, and after we open the file, we can see that we have achieved 100% sequence coverage based on the unique fragments listed in the table located on the left side of the screen. You can select or deselect certain digestion products while reviewing the results. A color coding scheme is used for highlighting the confirmed unique coverage, shown in green, versus confirmed ambiguous coverage, shown in pink, versus unique coverage possible, shown in blue. The coverage viewer results can be captured to clipboard for data export. Here we are comparing the coverage viewer results obtained for the same 100-mer single guide RNA digested with 4 different enzymes: RNase T1, hRNase 4, Rapizyme MC1, and Rapizyme Qusativin. While all enzymes achieved better than 80% sequence coverage, only MC1 and Qusativin were able to reach the full 100% coverage. However, we recommend using a combination of digestion enzymes in order to be able to generate a full sequence coverage reliably. In summary, I presented here a new workflow recently developed for LC-MS-based oligomapping, which enables full sequence coverage with increased confidence of assignments. 2 novel, recently launched RNase T2 enzymes, Rapizyme MC1 and Rapizyme Qusativin, have unique cleavage sites, and they have a great opportunity for creating overlapping digestion products compared to conventional RNase T1 digests. To improve the overall confidence in oligomap assignments and to increase the possibility for achieving a complete sequence coverage, we recommend using a panel of digestion enzymes. In the end, I would like to thank you for your attention.